takes a hell of a lot of money to start this process. So cell sizes are ideally small. Cell sizes are ideally small. Again, okay. Large surface area makes it really advantageous to exchange nutrients. Get nutrients in, get waste out. If we look on the next slide real quick, okay, here, just pretend you don't see anything below those green squares up there, okay? Don't read words, okay? We don't read words anyway, right? We just pick it up, we don't read the directions, we just start using stuff, right? So pretend you can't see it. So here is a large cell. It has a very large, this is that surface area here, okay? So I'm gonna have to govern this whole damn thing. That takes a lot of man effort. That takes a lot of energy to really monitor that whole surface area. Well, let's go to the very end here. What if I take that same square and now I divide it up, okay? Now I have much smaller surface area, right? This is much smaller than all of this right here, okay? I think we can all agree on that, right? So this is much more effective at getting stuff across really quickly, and it doesn't take a whole lot of effort. This, if I had to take something here and pump it all the way to the top of the surface here of that cell, that is going to cost a lot of ATP, and I need a lot of structure. I need a lot of structure to get it there. I need a lot of structure to maintain this huge ass shape. Sorry, huge shape. Okay, so having a very large cell is really not productive in terms of being efficient. It's not cost effective, and it requires a lot of stuff to even get it to that surface. All right. So cell sizes are ideally small. Why? To have that larger surface area to volume. Okay. A lot of surface area compared to that volume. 6 to 1. Almost 1 to 1. Okay. So I want a lot of surface area. And the body has this all over the place. Right? Whenever the body wants to increase the ability to absorb stuff, it increases the surface area. All right. It does this in the digestive tract. It does this a multitude of various different ways. We have plicae circularis, we have microvilli. We have a multitude of ways that it's going to help increase our surface area. Villi on plicae circularis, villi, and microvilli. So we have a lot of, so we have a lot more modifications inside the body. And you see that in multitude of various spots. I just say one in the small intestine there. All right. So to see all these suckers, we need a microscope. Duh. We have a multitude of various different scopes. All right. And the first one we have is a compound light microscope. Pretty straightforward, pretty easy. This is, basically what it looks like. All right. I wish we could get 200x on our scopes in our lab. The best that we can get is 100x. There's an oil immersion thing, and I know I'm talking gibberish and you guys don't really know, you probably don't really care, and that's fine, but I'm going to talk anyway. To make a clear image, but for the most part, we're seeing it on like 40x, really clear in our lab. And, so sometimes these images that they have in the book are fantastic, but we don't really get to see those, unfortunately. But this here is a typical light microscope, compound light microscope. We're using light. Okay. Light. Okay. It goes through a series of lenses and it focuses the images on our eye. Booyah, there's our eyeball. Okay. Goes through a series of lenses to help get this thing here, this image here. The magnification for this thing can be upwards of 1000x. Again, the ones in our lab is about 200 at the max with oil immersion, but we don't really get too much out of it because of our slides. The biggest thing that we have here, though, is what's called resolution. So we can get 0.2 micrometers away from the specimen with a compound light microscope, right? And that equates to about, 500 times better than the human eye. And I doubt, I wouldn't ask the 500 times better. I would ask more the distance between the specimen and the lenses, okay? And that's 0.2 micrometers, all right? That one I would ask. I just put 500 times better to show you, man, you can get really close to that specimen there. Really close, okay? And that aids the human eye tremendously. So now I can see, now I can see all these tiny little organelles that we have inside this thing here that we're going to be talking about here in a second. So this is the one that we use in our lab. What about other ones? We got a lot of other ones. We have an electron microscope, okay? And we have a couple different types. Transmission is what's on this slide, and it's shorthanded TEM. And you can use shorthand on an exam. I don't care. You have to at least list it out once, and then behind it, you can put it in parentheses TEM, if I was to ask this as a short answer question, okay? We have transmission electron microscopes. Now, an electron microscopes uses electrons. Those tiny little things that we talked about like forever ago, it seems like now, all right? Chapter, 2? Electrons. We pass those through a specimen instead of light. So because we're using, and electrons are really small, okay? So we focus all these electrons, shoot all these electrons through the specimen here. And instead of blasting them in your eyeball, because that wouldn't be conducive, we blast them into a television. And we can photograph that image. That same euglena that we see right here on the light microscope, on the compound light microscope, here's what it looks like with a transmission electron microscope. It transmits those beams of electrons through the specimen, okay? There it is. Pass through the specimen, all right? So here, light was being passed through the specimen, okay? But, it has limitations, okay? It's not as small as an individual electron. So you don't see all that detail inside there. You see a bunch of green gobbledygoo. You could probably make out a couple things, some chloroplast, maybe a little contractile vacuole in there, maybe some hairs on its surface or a little bitty flagellum here at the end. But here, now that I transmit those electrons, I can transmit those electrons through this thing. And since electrons are so small, I can get really close. From 0.2 to 0.0002, tens, hundreds, thousands, millions, micro. So I can get really great resolution here. This is about 100,000 times better than the human eye. So I went from the human eye being 1 to 500 times better using a compound light scope to 100,000 times better using a transmission electron microscope. So I increase my resolution. When I increase my resolution, I can get even closer. I can get even closer to that specimen. And now I can see a whole mess of organelles inside this little euglena. We have one other major type of electron microscope, right? And that's your scanning electron microscope. And again, shorthanded SEM. With SEM, now I can see, because I'm scanning, right? Because that's in its name. I'm scanning. When you scan something, you're just taking a glossary glance there at the surface. That's what you see here, all right? Light microscope, you can see a little bit of the surface, not a lot of great detail, because I don't have great resolution. And I see some bits and pieces inside, some of those organelles, again, not great resolution, so I don't really see in all the detail. Transmission electron microscope, I can blast those little tiny electrons in there and I can get great greater, obviously, resolution. And I can see the structures inside that little euglena. But as you can see there, I'm blasting it through. I don't really see the surface. And that's where we have the scanning electron microscope. All right. It blasts through there, puts those electrons through the specimen, puts it in a TV, and gets the image of, and that's the outer surface there of that same euglena that we looked at way back over here. Now you can see that little flagellum a hell of a lot more pronounced there, right? And now it looks all weird and creepy. Ugh. Okay. Same electron scope. Okay. I'm just changing how the beam of electrons are being bombarded on this thing, right? I spray the specimen with a thin coat of some kind of metal, and it depends, all right? But now that metal kind of gets scattered across the surface of it. And that's what gives me all of that detail that we saw right here, okay? They're a hell of expensive, take a lot to maintain. Obviously, that's why we don't have one, okay? But we have three major types. So, you know, over here, these are some of the kind of the big words here, and then I have them all on one slide so you can kind of see them all together, okay? And you can kind of see the difference. You can get huge amount of magnification there, okay? 200x, you can see this is 1500x, this is 12,500x, you all can read. But you can see the amount of magnification I can do by changing light to penetrating electrons to dispersed electrons just getting around on the surface of it, okay? And I keep using that word resolution, okay? And resolution, you know, these words is what I was saying, get really, 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 really close, okay? Resolution is just the minimum distance between two objects, okay? Again, microscope, I can't get as close because I'm using light, all right? It's a little bit bigger than electrons. With electrons, I get super close. So I have huge amount of resolution there, okay? 100,000 times better than the human eye. So I'm able to get really close and I can see in a lot of fantastic detail. Magnification is just the ratio between the size of an object, an image, and its actual size. In electron, I'm able to magnify that a heck of a lot more because I can get really, really close, and I can have fantastic resolution. So it's able to magnify the image a heck of a lot better. Contrast. Contrast is just the difference in shading that occurs with an object when you coat it with something and compare it to its background, and that's what you see there with that scanning electron microscope. I've coated it in some metal spray. Excuse me. And that way, those electrons kind of bounce off the outside surface of that instead of penetrating through, and I'm able to kind of see great contrast with them. This is what we do in the body. You'll fluorescently tag molecules, watch where they go. You'll irradiate tissues. You'll chug that barium, watch it wash down through your digestive tract, and you'll be able to see, you know, if you have any ulcerations or any place where you have increased uptake of that barium and increased activity, to maybe point out, you know, potential issues, cancer, tears, anything along those lines. So contrast is used quite a bit in the medical field. All right. I could ask this as a short answer question, you know, maybe compare and contrast compound light microscope scanning and transmission. You know, that makes a good short answer question some semesters. Sometimes it doesn't. It just depends on my mood. But that does make a good potential short answer question. So now that we got a scope, now we can see all the little bugs that are flying around us, all right? So we're gonna start with prokaryotic cells first. These are the basic bacteria, all right? We're gonna spend the vast majority of the time with eukaryotic cells here. So prokaryotic cells. The big thing about these guys is that they lack a membrane-bound nucleus. They don't have it. We do, okay? That's one huge difference between a prokaryotic organism, bacteria, and a eukaryotic organism, animals, us, okay? Plants, all right? We have that membrane-bound nucleus. Because of that one little thing there, it is much simpler for prokaryotes, bacteria, to replicate, divide. It's much more complex. In us, because we have that we have that membrane-bound nucleus, okay? We have a lot of the same things in a prokaryotic organism that we do. It's just a heck of a lot more simpler. So that's why we kind of separate these two things out. Thing to remember, who's a prokaryote? Bacteria and archaea. All right, bacteria, all the nasty little bugs that cause you to be sick. They also make some drugs, penicillin, right? They also make some stuff in industry, from cleaners. They help make bread. So, they do a lot of fantastic things, as well as cause a lot of issues on us as well. Archaea was that one that's more closely related to us. They live in those extreme environments, those extreme habitats there. They do have some nuances and some difference to them. And if you're concerned or interested in that, I should say, take micro, okay? Because they'll dive into that more there. We're just going to keep it really simple here. When we look at the shape of a cell, of a prokaryotic cell, it varies, okay? It varies. When we look at eukaryotic organisms, it's going to be two choices, night and day, okay? It's either blobby, okay, or it's stiff and rigid, okay? With prokaryotes, we have, as you can kind of see here, a number of various different shapes. Coccus means circular. Bacillus kind of means box-like. Okay. And spirillum, like a spiral, okay? Squiggly. You may see it listed in some texts, and I'm sure you're really concerned. Spirillum or spirochete. We have both of these that we would have looked at in lab, and you would see some of the difference between them. And a spirillum, as you can kind of see, is a little bit more kind of long-winded in its shape. It's a little bit more rigid. Spirochetes are just a little bit more flexible, but semantics, who cares? 3 different shapes is what I want you to know. Circle, coccus, square, bacillus, squiggly line, spirillum. Okay, those are your 3 major structures here. They occur in one of those things. They can be singular, as you can kind of see out here, bacillus by itself. And here you can see it, they can be all strung together as well, right? And this is significant in going to diagnose, Streptococcus pyogenes, Streptococci, the specific shape of these things does allow the medical community to identify it because they'll all have a certain kind of shape to it, either singularly or all stuck together like that. So it is significant if you go in that route. When we look at the plasma membrane here, I will say this, okay? There are a multitude of various different names for this. When we talk about the cell membrane, we are talking about this, okay? Move those post-its out of the way, all right? We are talking about this. Plasma membrane is also cell membrane. Okay, so plasma membrane is cell membrane. What's in that thing? Well, fats. It is a lipid bilayer, okay? A phospholipid bilayer. Got that phosphate head, got those lipid tails, As you can see, we also have proteins embedded in this thing. Sometimes they're on one side. If they're only found on one side, they're going to have a name. And this is getting into a little bit of what we're going to talk about in chapter 5, but knowledge never hurt nobody, right? These are both peripheral because they're around the outside. Okay? This one spans the entire width. It's integrated within this phospholipid bilayer. Integral. Okay? So we can have these plasma membranes scattered all over. Okay? So cell membrane, plasma membrane is this phospholipid bilayer. Okay? Is this phospholipid bilayer. It can have proteins scattered around. Also, in a bacteria, they may have a cell wall. This helps to maintain that kind of rigid shape of whatever the organism is, okay? Bacillus. Function, maintain shape. Defines the thing, gives a lot of strength. It can survive a lot better with a cell wall around that plasma membrane, okay? Because it gives them strength, all right? Protects them a little bit. Excuse me. Bacteria may also have this thing called a glycocalyx. What do we call it? We usually call it a capsule. Okay? We usually call this thing a capsule. This is poly many. Poly means many. And saccharides are sugar. So this is a sugar coat. Okay? This protects them even more, okay? Because it's this kind of wet, thick, kind of outer layer around everything that allows these bacteria to stick to the surface of something for like months on end. And that's why you should, cover your mouth when you sneeze, right? And if you do sneeze in your hand, go wash those suckers, man. Okay, because now it's on your hands, and now you're going to fondle everything. Fondle that pin, and stick it in your mouth. Blah. Okay? So that glycocalyx allows these things to survive on surfaces for a really long period of time. Okay? And there you can see the capsule, the cell wall, the plasma membrane. All three of these things are collectively that cell envelope. We call it a cell envelope. I call it a cell envelope because I'm cool. All right. So 1, 2, 3, from innermost to outermost. All three of those things are called the cell envelope. All right. And you can kind of see some of them have little fimbriae, little hair-like things. They even allow even greater adhesion to a surface, so it doesn't just slide down. Okay? It can really be anchored there to it. All right. So there's a lot of adaptations that we have here on this outside surface of a prokaryotic cell, okay? And we'll do a deep dive in chapter 5 into what all of these outer parts and pieces are of the plasma membrane. Just doing a kind of a brief intro to it right now. So if we look at the prokaryotic cell really simply, we have this stuff called a nucleoid region. Okay, region containing the DNA, and it's circular. It's weird and bizarre. Okay, ours are those wonderful little X's, right? Well, it starts as something else prior to those X's, but it's not circular. All right, prokaryotic organisms have a nucleoid region. Remember, they don't have a defined nucleus. All right, if I draw a cell right here, and then in the center of that cell is the nucleus, right? You know that from high school. There inside that little cell, or that little nucleus of that little cell, is our squiggly line of genetic material. In bacteria, okay, they don't have a nucleoid region, or I'm sorry, they don't have a nuclear membrane, a defined nucleus, right? So we have all that squiggly line just sitting there kind of in the center of the cell, okay? And it's all circular in nature, but it's all gobbledygooked up in the center. Makes it hella easy to really get in there and copy this thing. That's why bacteria can, replicate and divide every, 12 to 15 minutes. Very easy to do. Plasmids are accessory structures. They're kind of extra chromosomal. They do a number of different things. You'll notice I didn't highlight it. Ribosomes. Ribosomes allow for protein synthesis. So we'll see the same thing in us as well. And you see them here. Again, both of them pretty much have some basic structures associated with them. It's just they're much more basic here in a prokaryotic organism. So the two kind of biggest, and they have a semi-fluid, solution. So they have some cytoplasm within them. So do we. It's just a watery medium that is within the plasma membrane. And these two major things that I would like you to know: a nucleoid region, because it's not a centrally defined nucleus, no plasma membrane. That's where the circular DNA is found. We'll be talking about this in 2 units from now when we talk about how cells divide, how they read the DNA, and how they copy that DNA. So we'll talk about this nucleoid region a little bit later. That's why I want you to remember it now. The other thing that we'll talk about in eukaryotes, as well as in that unit, are ribosomes. And ribosomes help us to read the DNA. Okay, copy the DNA, do all that fun stuff that we're going to be talking about in mitosis and meiosis later on. Externally, there's some stuff that jut off that plasma membrane, or really that glycocalyx, that capsule. We have a flagellum. It's just a, if I look at the cell here, okay, there you go. It's the flagellum. It provides motility. It's how this thing can move around in a watery medium. You'll see it in sperm as well. Fimbriae, you saw those there on that hair, on this previous slide here. They're just little hairs, okay? Bristle, okay? And it allows this bacterium to stay there and not get moved away very easily. The other thing is this really disturbing thing called the conjugation pili, all right? It's just a rigid tubular structure, and it's how it injects its genetic material into an unsuspecting cell, okay? So that's how these bacteria get other cells to do their bidding, right? This conjugation pili. Pilus is singular. It injects it into another cell, our cells, when the bacteria gets inside of us. It takes this conjugation pili and starts popping our eukaryotic cells, filling it up with their genetic material. And now that genetic material gets incorporated into that cellular nucleus. And now that cell becomes a manufacturer of bacteria. That's how bacteria thrive. Okay? So these are the things that I have highlighted. So please remember those. They're probably important for the exam. All right. So that gets us into, let me do something here.